Hello, my name is Kristina Pickering. I am a junior at Notre Dame of Maryland University, and today I will be presenting my research entitled The Analysis of the Genomic Sequence of the Gromphadorhina Portentosa and its Microorganisms. Another name that is being considered for the title is The Preliminary Characterization of the G. portentosa, cockroach genes mediating experience-dependent neuronal plasticity and gut microbe analysis. The purpose of the experiment is to analyze the Gromphadorhina Portentosa, otherwise known as the giant Madagascar hissing cockroach, to understand neuron function and what genes or proteins could play a role in neuron function. This project entails two individual lab projects that looks to 1. Knock down the supposed neuron functioning proteins using siRNA, otherwise known as small interfering RNA, while 2. Also analyzing the microorganisms inside of the G. portentosa. At this moment, I do not have any extracted DNA sequences to make the siRNA, so the first big project is to continue to attempt to identify, using primers found in literature, to look for certain genes of interest to see whether or not these genes are present in the cockroach, and then send the DNA away to be sequenced. Once the sequenced DNA comes back, I will then be able to design specific siRNA molecules that will interfere with the DNA I have sequenced and knock down the mRNA levels of protein. With this information, I would then be able to look at neuron function, which is the main interest of my lab. The second lab project conducted in conjunction to Part 1 is looking at the microorganisms in the gut of the G. portentosa. Here, I am using similar techniques such as using certain types of primers found in literature to identify certain types of groups of bacteria or genera of bacteria present in the G. portentosa. My project is therefore a methods-based one, where the goal is to ultimately have an impact on the neuroscience projects that are taking place in my lab. In my project, I am learning methods such as DNA extraction, how to optimize temperature and primers for PCR, which are two methods I have primarily used so far. To give background, DNA extraction involves dissection of the cockroach, looking at the head, gut, and the legs specifically, and extracting DNA using methods such as homogenization and colony extraction. Once I have collected the DNA, I then add primers in PCR, which are single-stranded nucleotide sequences that are used to find a specific sequence it is coded for in the DNA and attach itself to it. The process of PCR, which stands for polymerase chain reaction, utilizes these primers by breaking the double-stranded DNA apart, adding primers which attach to the sequence it codes for, and then copying the DNA until its end where both strands can be placed back into its double-stranded sequence. This allows for numerous copies of DNA to be made for genetic analysis. I have also utilized bioinformatic tools such as DNA Subway and phylogenetic mapping that use software programs to do further analysis on the DNA I find. When I am done identifying the primers at work, I plan to move to the next step for both of my projects, which is taking the extracted DNA to get sequenced while looking at the specific primers that were used to find the DNA sequences. Using specific primers in the PCR extraction, I will then take the PCR product and place it into a gel electrophoresis, where the bands based on size of genetic material present will be separated with an electric charge to see the bands that hold the DNA sequence that will then be sent off for sequencing. Once a sequence is found that is consistent with the literature of the gene of interest, a siRNA will then be designed to see if the lab I am working at that deals with experience-dependent changes in neuron activity, whether or not that protein being looked at will be knocked down to produce results. There is a chance nothing will be seen, as a protein might be compensating for the one altered, or siRNA treatment may block the experience-induced change in neuron activity. As this is a methods-based project, I have begun the long process of collecting and sequencing DNA as shown through my results. For the first part of my lab that looks at the G. portentosa for siRNA designing, it can be seen that I have been able to obtain DNA that is mostly cleared of any contaminants, as shown with the low 260, 230 values, and mostly pure, shown with the 260, 280 values that are below 2. The graphs are a physical representation of the amount of DNA that I have been able to collect. The high peaks indicate a relatively large amount of DNA collected. The gels found here from the gel electrophoresis with my original choice of cytochromoxidase primers have been able to produce results, as shown from the second gel. However, these results are not similar enough to the ones found in literature to be able to sequence. The next two slides show two of the programs that I am using for DNA analysis, DNA Subway and phylogenetic mapping. Both tools are used to determine how similar the genetic sequence of the G. portentosa is to other organisms. The data shown was an exercise I conducted to use the programs, as my data is not consistent enough with the literature to do further testing. The next step would be to use these tools to map the genes I am looking at to see how familiar they are to genes of similar interest in the literature in other species, such as the Periplaneta americana, to build a phylogenetic tree to depict any similarities or differences. In the second part of my experiment, where I began analysis on the microorganisms, I used media plates such as sheep's blood plates and agar plates in anaerobic, aerobic, and microaerobic conditions to grow colonies that were then broken to collect DNA. This DNA then had primers added to it from 3 different categories found in the green graph. Eubacteria that looks at bacteria, Eukarya that looks for eukaryotes and protozoans, and Archaeobacteria that looks for archaeans. Essentially, I am looking at the 3 main domains of life. PCR was then performed along with a gel electrophoresis, which, as shown by the following gels, did not produce viable results for sequencing. I am currently looking at optimizing PCR temperatures and primers to look for possible sequences that are similar to the ones found in literature. When I am done with both parts of my project, my results would be able to assist the lab I am in identify certain genes or proteins that may be playing a role in experience-dependent neuron activity, as well as information on the microorganisms that reside in the G. portentosa for other possible experiments. At this time, I would like to thank all those in lab who have helped me thus far. Thank you to Dr. Sosa, who's my professor, Esther B., who allowed me to continue her project after graduation, and Mr. Pat Bell and Mauricio, who have helped me find my way around the lab. Thank you for listening. I hope you enjoyed my presentation. I will now